What major group are we talking about? Prokaryotes, right? Who are those? Yeah, bacteria and archaea. So we spent a good amount of time going over this chapter already, and we're mostly finished. So we're just going to hit on the last couple of slides. Remember, we talked about the evolution of the group. We talked about where you find them. We talked about historical atmosphere. We talked about structures. This was all just labeling stuff. You guys remember that. This was two lectures ago. And then you've got the whole section that we spent talking about. Oh, this was a good one. You guys remember this, right? Comparison of features that bacteria and archaea share in common and that archaea and prokaryota share in common. And we talked about this, relative complexity. Anybody want to sum that up? Who's feeling brave? Not so much on the Monday at 11, huh? Remember, we talked about that you guys don't have to memorize these characteristics of each group, but rather understand that as you see those derived traits that you see come later, right? What's an ancestral trait versus derived? What's ancestral? You guys remember that? What does it mean for a trait to be ancestral? Yeah, it's been passed down through generations. It's an older trait, right? It's a trait that you find further back towards the root of your phylogenetic tree. If we're looking at this relationship between bacteria, archaea, and eukaryota. And then contrast that with a derived trait. What is a derived trait? Kind of the opposite, right, of ancestral. That what? That you developed? Yeah, it's just later developed, right, or evolved. So if you have groups that share derived traits, it means that they have accumulated those traits later in evolutionary history, right? So we were just talking about the difference between sort of the simplicity of the traits that are shared ancestral traits that would have been there first. Things like the single circular chromosome, right? One type of RNA polymerase and other enzymes. Things that are more simplistic. And then when you see the evolution of the group that leads to eukaryotes, right? Archaea and then eukaryotes, you see those traits becoming more complex. Things like introns in the genes, multiple types of enzymes used in DNA replication. What else do we talk about? Histones, right? So then increasing complexity. So it's that theme that you guys need to get, right? And it's not these individual traits that you're going to have to actually explain or describe. Make sense? You guys good with that? Okay, so that was one of the trickier points that we talked about. We talked about gram-negative versus gram-positive as a way to identify groups of bacteria. We talked a little bit about gram staining, horizontal gene transfer. Yes, 3 ways to do that. So make sure you're comfortable there. Metabolism, we talked about 4 different types, right? Where you get your energy, where you get your carbon skeletons. Basically what it comes down to, yes? You guys remember all this? It's all coming back to you. I know it's been 5 days since we last talked about prokaryotes. There's your flow chart. And then we spent a good deal of time talking about pathogens, right? We talked about all the bad things that prokaryotes can do, largely bacteria. Why don't we focus so much on archaea when we talk about pathogens? Yeah, there's none that have been identified conclusively to cause pathogenicity in other organisms at this point. Could it possibly still be determined that that is possible or the case? Yeah, so research is ongoing in that area. We talked about MRSA, we talked about food poisoning. So we ended on sort of all the negative things, right, that impact us that prokaryotes can do, but then I promised you we'd pick up here. Beneficial bacteria, okay, so we're gonna swing it back into the direction of positivity a little bit. So they're not all bad. In fact, when you start looking at bacteria, it's a really small relative percentage of species that are actually problematic or pathogenic, right? Why do the ones that are pathogenic get so much attention, do you think? Because they're pathogenic, right? If they're causing disease or death or trouble or problems for the human population, they're going to get largely the attention, right? Medical research and things like that. So it's often overlooked because there are lots of beneficial prokaryotes as well. So we're going to look at some of those benefits. We'll start at sort of the ecosystem level, and then in the next slide we'll talk about human-specific benefits that we can find with prokaryotes. So remember, without prokaryotes, life on the planet would not be possible. Why do I say that? Go back a couple of lectures and think about cyanobacteria. What happened when cyanobacteria, or the ancestors of cyanobacteria, evolved the ability to photosynthesize? Yeah, changed the atmosphere entirely from anoxic, right, to an oxygen-rich atmosphere that allowed for the evolution of all aerobic life to come after that, right? So really important in that sense, and we talked about that already. Another area where you see benefits of prokaryotes is in nutrient cycling. So we haven't talked about nutrient cycling probably at all yet. We'll talk about it a lot when we get to the end of the semester and we're talking about ecology, right, and ecological relationships. But we're talking about nutrient cycling. We're talking about the fact that organic materials are cycled within an ecosystem. Right? How does that cycling of nutrients happen? How are organic materials returned to the system? What do you guys think I'm talking about? Two things, basically. You've got waste, right? Waste from living organisms. And you've also got dead organisms, right? So you've got decomposition. When you have these large macromolecules, let's say a tree falls in the woods, right? The old saying. When it starts to decompose, what is actually happening? You ever think about the process of decomposition? So you've got this tree that spent however, let's say it lived 100 years. It spent 100 years putting together molecules, right? Assembling tissues, building things like bark and leaves, right? All the tissues that make up that tree. When it's done, with its usable life is over and it falls down, something has to happen to all the bonds, right? When you're building tissues, you're making chemical bonds, yes? Everybody's good with that? Remember that? So you've got to have organisms that are capable of breaking those bonds in order to cause decomposition to progress. Without that decomposition, that energy and those nutrients that are built up into those tissues sits there forever. Okay, so decomposition is a huge part of cycling nutrients back into the system. Okay, so bacteria and archaea are big decomposers. So a huge role in ecological nutrient cycling. And then nitrogen fixation. You guys may or may not have heard of the term nitrogen fixation before, but I know you've heard the term carbon fixation. Does that ring a bell? What am I talking about? You remember where you've heard of carbon fixation before? You know I'm always asking you to remember stuff from the first half of biology, right? There's somewhere back there. How about photosynthesis? Remember that? Have you heard that term before? Carbon fixation? No? Yes? Give me a thumbs up if you have. Carbon fixation. Some of you have. Okay. I'm not going to pick on you. I'm just taking a poll. So anytime you see the term fixation in terms of an element like carbon and nitrogen, what you're talking about is taking an inorganic form and converting it somehow into an organic form that's usable by a living thing. Okay, so in this context, that's what we're talking about. When you fix carbon during photosynthesis, you're taking inorganic carbon dioxide from the atmosphere and you're modifying it, right? That photosynthetic organism is modifying it chemically to make it into a compound that it can use, okay? So the same thing happens with nitrogen. You guys ever study the composition of our atmosphere as far as gases? Do you know what the most abundant, you're nodding, what's the most abundant gas in the atmosphere? Nitrogen, right? And it exists as N2, okay? But we can't use N2 in that form. That's inorganic nitrogen gas, okay? So what nitrogen fixation means is converting atmospheric nitrogen to other molecules that are usable by living organisms. Most of us, most living organisms can't do that innately, okay? So we depend upon other organisms that can do that to fix that nitrogen into usable form. So there are plenty of examples of nitrogen fixing bacteria. When we get to our plant chapter in just a couple of weeks, we'll talk about the relationship between those soil microbes that can fix nitrogen and the roots of plants. Why is nitrogen important to plants? Any gardeners in the room? Anybody? Sorry? Soil. Say it again. Nitrogen soil. You need nitrogen in the soil. Yeah. When you put fertilizer down onto your garden, what's the #1 ingredient in that fertilizer? You guys know? Usually nitrogen or some sort of nitrate or something along those lines. Nitrogen is important for building things like protein and DNA. And it's really important, often can be the limiting nutrient for plant growth, okay? So it's really important that plants can get enough nitrogen, but plants are like us. They can't fix nitrogen. They can't use nitrogen from the atmosphere. So they rely on bacteria that live in the soil that can do that do it naturally as part of their metabolism. They change that atmospheric nitrogen into forms that plants can then use, uptake, build proteins, build tissues, build DNA, and then you have the base of the food chain, right? Essentially. Okay, so nitrogen fixation is really important. Not because you reach into the soil and eat nitrogen, right? But because all of our food comes from photosynthesizers, and photosynthesizers, like plants, largely need that nitrogen fixation to happen. Right? So we'll get to that a little bit more in detail when we get to plants, like I said, but that's a good sort of introduction to that ecosystem level benefit. So they make oxygen, they decompose organic materials, returning those nutrients into the system, and they fix nitrogen for the rest of us. Okay? That's better than thinking about things like MRSA and food poisoning. Right? I told you about all that. Okay. All right, let's talk about bringing it a little bit closer to home, shall we? So food production, human gut microbes, we talked about this a little bit. These are all human-specific benefits. So it gets interesting when we start talking about ourselves, right? We always like to discuss impact on humans. So how about food products that rely on prokaryotes for the production of those products? What about cheese? Who likes cheese? Yeah, like a main source of protein in my particular diet. Yogurt, vinegar, sauerkraut, cured sausage, lots of things. The list goes on and on. Things that are fermented, right? Things that are cultured. So this practice goes back at least 4,000 years ago. This number, 4,000 years, that's the number in your textbook. I've read some papers recently that bring that number back even further, like 10,000, 12,000 years ago, the first evidence of cultured dairy. So human populations that are keeping animals and using their milk and doing things with that milk, turning it into things like cheese and other cultured products like sour cream type stuff, right? So that's pretty cool that that's been going on for a while. All right, human gut microbiota we talked about. What is your microbiome? Remind me. What are we talking about when we talk about your microbiome? Talk about the Human Microbiome Project, right? Who are you looking at? Yeah.
There's all the bacteria.
Yeah, they're all of the microbes that live on and in you. Remember, we said you're at least as many microbial cells as you are human cells, maybe more, depending upon the source you look at. So you've got all of these microbes. We're specifically talking about gut microbes here in this example. Those are those bacteria that live in your intestines. Are they helpful or are they harmful to you? Most of the time helpful, yeah. At the very least, they're just kind of there, right? For the most part, they're not doing any harm, or they might be helping you. So how do they help? They can help with digestion and absorption of nutrients. There are certain things that we're just not that efficient at getting out of our food, but there are microbes that live in your gut that are better at it. And they can break stuff down and sort of contribute to that absorption process in your own intestines. There are certain gut microbes that produce vitamin K. You can't produce vitamin K yourself, but you need it. One very important reason that you need it is for blood clotting. What is blood clotting? Yeah, you gotta have it for wounds to close, right? If you cut yourself and you can't clot, you will bleed to death from a paper cut, right? So you gotta be able to clot. There are a lot of chemical processes involved in the cascade that is blood clotting that ends up with you having a scab, essentially, right? One of those steps requires vitamin K, and one of the places that you can get that is from these microbes that live in the gut. So that's a symbiotic relationship. Let me pop down here a minute. We've talked about symbiosis. We're going to talk about symbiosis a lot more going forward. Okay, we're talking about evolution of eukaryotes through symbiosis here shortly. And we've mentioned these terms before too, so I want to call your attention to these. These are important. Mutualism and commensalism. Yes, you guys remember talking about this? Mutualism is a mutually beneficial relationship. Both species involved in this symbiotic relationship, or this living together in close association, are benefiting in some way. Right? You win and they win. That's mutualism in symbiosis. Commensalism, remember, is the term for the whole one species benefits, but the other one is lifetime. You're here, you're not bothering me, it's fine. That's commensal. Okay, one partner benefits and the other is unharmed. So some other things that you've got going on here with immunity, with your gut microbes, training and maintenance of your immune system. So your immune system, we've talked about to some extent during the virus chapter, and a little bit in this bacteria chapter, works by recognizing foreign versus self, right? If something foreign appears, your immune cells are trained to recognize that, but they're not trained from birth, they have to learn, right? And so there's some evidence that those microbes that are in and on your body surfaces contribute to that training and maintenance of knowledge of self versus non-self. And then this one is really interesting. Protection against pathogenic microorganisms. So as Grace said, most of the time, the microbes that live in your guts are causing you no problems. Yeah? Either they're mutualistic, they're helping you in some way, and you're providing them with shelter and food and a nice warm place to live, or they're commensal. From time to time, you get a mutant strand that can become pathogenic, or you ingest something that can be pathogenic, okay? But if you have a healthy gut flora or a healthy gut microbiota, they're going to protect their turf, all right? So the idea here is that opportunistic pathogens can come in, and if you have sort of holes in your coverage, if there's available real estate, so to speak, so I'm talking about your intestines, if there's room for pathogenic microbes to get in and cause problems, they might do that. But these non-pathogenic symbiotic microbes that live in your gut will protect you, not necessarily because they just like you so much, but they're protecting their home turf, right? They're competing for resources, and they can often outcompete the invader, okay, which is good for you. So it protects you against invasion by potentially pathogenic microbes. Does that make sense? So there's a lot of things that these guys do for you. The other interesting thing about learning about these microbes in your microbiome is that we really don't know the extent of the benefit of having this healthy flora. Okay, we talked a little bit about probiotics. You guys remember that? We brought that up. You can actually take tablets to sort of seed your microbiome with new colony forming units. It's available. The literature is conflicting, whether it's good for everyone or if it's just good in certain cases, like when you really need it, maybe after a round of antibiotics or for certain types of digestive issues like irritable bowel syndrome and things like that can sometimes be treated by it. So what I'm saying is, like, everyone doesn't need to go to the grocery store and buy probiotics. Probably won't hurt you, but it can get expensive. So maybe if that's something that you are interested in, go on the advice of a physician or a nutritionist or something along those lines, because it may not be necessarily evidentiary support for help for your particular condition, but it's an option. Questions about this stuff? Microbiome, how it helps you. You guys are very excited about the microbiome. You can tell. Okay. Last slide for this chapter. One more benefit, and this is larger scale. Okay, this is another application, a little bit more specific. We're talking about bioremediation. Have you guys heard that term before? Anybody know what I'm talking about? It's bioremediation. If you remediate something, you're sort of going back and fixing a problem, right? So bioremediation means using natural systems to clean up messes, right, in a nutshell. When we're talking specifically about microbial bioremediation, we're talking about using bacteria and archaea to do that. We're using prokaryotes and their naturally occurring metabolic processes to help us clean up things like pollution, okay? Some good examples of doing this are things like taking pesticide and fertilizers, toxic heavy metals, and oil out of the atmosphere. Okay, so there are different prokaryotes. Remember how we talked about extremophiles? So these guys are doing things. Bacteria, archaea, all these different species are doing things that seem counterintuitive to being healthy, right? Like if you ingest a whole bunch of selenium and mercury, that is bad for you, yes? That is not what you want to do, but there are microorganisms who thrive on that kind of thing, right? And so we can harness those naturally occurring abilities to help us clean up water and soil where that stuff exists, where we don't want it, right? Because it can be harmful to us. So this is a nice little example here. of microbes that can clean up oil spills, as I mentioned. So there are certain strains, lots of different genera of bacteria can do this, where they metabolize hydrocarbons. So when you guys think about petroleum products, oil, things like that, those are largely hydrocarbon chains. Remember that from the macromolecule unit way back in Bio 1, talking about fat. They've got largely just chains of hydrocarbons. So if you've got bacteria that can break those chains down into smaller pieces, that helps to remediate, yes, bioremediate, something like an oil spill, making those molecules, those oil molecules, smaller and more soluble in water. Okay, so either they're breaking up and they're getting to be less sort of adhesive to one another, or they're even metabolized into less impactful molecules, ecologically speaking. Okay, so there are, like I said, lots of genera of bacteria that can do this naturally. So you can take those bacteria and actually use them to clean up something like when a ship spills oil into a gulf, for example, right? Or even freshwater ecosystems. There's evidence that this works there too. There's an interesting story. I should link to the Supreme Court case because it's actually pretty cool. So the first patent application for bioremediation in the US was for genetically modified oil-eating bacteria. So what this guy did was he went and took multiple species of bacteria that can digest these hydrocarbons, and he took those genes, independent examples of those genes that all code for that ability, and put them into one population of bacteria. So basically like a super bacterium that could digest hydrocarbons in different ways to help with bioremediation. And the interesting thing about the court case that went all the way to the Supreme Court to show that you could indeed patent something that is alive. Kind of an interesting story. I think what I'll do is go back into this slide and just link to this to an article about that Supreme Court case, because it's kind of interesting. Nice tie into sort of politics and the legal system and patent law and bioremediation and prokaryotes. That makes sense? All right, so it's not all gloom and doom. It's not all strep throat and bubonic plague and salmonella, right? Some good things too. All right, so this is the same pattern that we'll see. Remember, each chapter is gonna look just like this: introduction, characteristics, evolutionary history, notable examples, benefits, and harm. Okay, so that's pretty much the nutshell of the design of the chapter that we'll see going forward. All right. So that finishes up prokaryotes. The next chapter we're going to get into is on protists. Okay, but before we do that, I want to watch a video. I posted this video link in your D2L shell, but it's under the virus chapter because it's more closely related to that. So you guys can go back and watch this, but we're going to watch it now together because I want to discuss a few things. It is about a pandemic. That's relevant, right? That lasted 15 million years. How long have we been in the middle of the current pandemic? Going on a year, right, since it's really been impacting us here. How do you guys feel about 15 billion years? I don't think so, right? Gives me anxiety. It's actually really interesting. So it is this video, it's about 10 minutes long. I'll stop it a few times and we'll talk about stuff. But what we're going to look at here is some review about what we talked about in the virus chapter as it relates to genome evolution and horizontal gene transfer. It's also talking about pandemics, which we talked about in this chapter and the virus chapter. So it's kind of bringing it all together, but in a really cool, applicable way. All right, so let's watch this and see what we come up with here.
Around 33 million years ago, in the late Oligocene, a new virus emerged and infected some kind of man. As we're all well aware at this point, viral infections happen and spread all the time. But this time was different. It would turn out to be the beginning of a viral outbreak that left its mark on.
Does it sound okay? It's kind of echoey. Can you guys hear it all right?
The genomes of almost every order of mammals around today. The start of a huge ancient pandemic, one that touched many different species, spanned the globe, and lasted for more than 15 million years. Yes, now imagine a pandemic that lasted 15 million years. And we know this pandemic happened because in 2001, biologists discovered part of this ancient virus trapped in our DNA, a molecular fossil entombed in our own genome. This fossil wasn't the actual virus itself, but a fragment of its genetic material. Scientists named this DNA sequence ERBFC, and it's just one of a growing number of viral fossils that have been discovered in the genomes of modern animals. And I know you have questions about this because I definitely do. Like, what was this virus? How did it end up in our genome? What disease did it cause, if any? And is it doing anything in there right now? Well, we don't have all the answers yet. The legacy of this ancient pandemic is just one of the many stories hidden in our very own DNA, just waiting to be discovered. Whether they're animals, plants, or fungi, all living organisms have at least one thing in common. Their DNA is riddled with the remains of ancient viruses, and humans are no exception. These viral leftovers are known as endogenous retroviruses, or ERVs, and most of them are the remnants of ancient retroviruses that infected the egg or sperm cells of our mammal ancestors. Those kinds of viruses insert themselves into their host DNA as part of their normal life cycle. And when this happens in reproductive cells, that viral sequence can be passed down from parent to offspring. Now, this doesn't happen with every viral infection. In fact, viruses embedding in the genome of their host is pretty rare because everything has to go just right for this to happen. First, the virus has to integrate into the DNA of the sperm or egg cells at just the right spots to not kill those. Those cells have to be the lucky few that actually end up forming a fertilized egg, which is what allows the ERV to be passed down to the next generation. And then it has to keep getting passed on, even as these organisms change and evolve over time. But over millions of years, these rare events have happened often enough to leave a viral fossil record behind. In some way,
You guys see that? That's a tree we can handle, right? You guys wanna look at this one? No, I'm not gonna make you. But one of the things I want to point out here is that this also harkens back to our discussion on phylogenetic trees and the applicability of phylogenetics. Okay, so we're talking about the first part. He was introducing how you see remnants of viral genomes in the genome of other organisms, including us. Right, we touched on that a little bit. He mentioned that the viral DNA would have to be inserted into a particular type of cell. Did you guys catch that? In order for it to be passed down from the organism who was initially infected with that virus to subsequent generations, what kind of cells does that have to get into? Sperm cells and egg cells, or I should say, right? So in order for something like a I guess you could call this like epigenetics almost, right? Like new DNA being introduced into the organism that's living. So this kind of goes against what we talked about as far as inheritance of acquired characteristics. Right? Remember how we said that Lamarck talked about the giraffes and how that was so kind of goofy based on what we know about inheritance patterns? But I said he wasn't entirely wrong. We're going to say he was wrong, but he wasn't entirely wrong because the things that we're learning now sort of teach us new things about how you can sort of pass those traits on. This would be kind of an example of that, right? Where you get a viral infection, and then remember how viruses work, right? That they actually are introducing their own core, that viral core, DNA or RNA, into your cells, and then interacting really closely with your own machinery, your own cellular machinery, including your enzymes and your nucleotide pool and all of those things. So you can see where that window of opportunity exists, right, for pieces of DNA to get into and out of genomes. So basically that's swapping, but it would have to happen in a cell that goes on to be a reproductive cell. Okay, so keep that in mind. Good? Sorry, this sounds a little distracting.
Often enough to leave a viral fossil record behind. In some ways, it's actually a lot like the other kinds of fossilization I've talked about around here. It requires specific conditions and only happens some of the time. Nonetheless, ERVs are so common in our DNA that, by most estimates, they make up around 8% of our total genome. And that's about 4 times higher than the proportion of our genome that's made up of genes, the sequences that actually code for proteins. So yes, you heard that right, but just in case you didn't, I'm going to say it again. In terms of DNA content, we're about 8% virus and 1 to 2% genes. So what I'm saying.
What? I already told you that you're at least half prokaryote, right? That you have as many prokaryotic cells living in and on you as you do human cells. But now he's saying that you are 8% viral DNA. That's crazy to think about, right? And 1 to 2% genes. What is everything else in your genome? You guys remember? This would be from the end of Bio 1, probably talking about genetics. What is all that stuff? We talked about introns, right, a little bit in here. So you guys are familiar with the idea that you have non-coding regions? Yes? Ring a bell? Stop, give me a thumbs up if that rings a bell. Non-coding regions, right? So things like regulatory elements, things that turn genes on and turn genes off, right? Promoters, those things that don't necessarily code for proteins but are there for other purposes, or the stuff that we used to call junk DNA, we don't necessarily refer to as junk anymore, we just don't necessarily know what it does, those non-coding regions. So you're only 1 to 2% genes, the rest of it's either non-coding or viral DNA. It makes you think about who you are a little bit, at least evolutionarily.
You are more virus than you are genes. Now, figuring out where each bit of that viral DNA came from isn't easy. Many of these sequences have built up so many mutations and have been shuffled around so much that they're really fragmented and hard to make sense of. But in the case of ERBFC, that sequence tells the story of an ancient, global, multi-species pandemic, or maybe more accurately, a series of epidemics played out over a huge span of time. And while ERBFC was first identified in 2001, it wasn't until 2016 that a group of biologists at Boston College was able to piece together the history of that particular ancient virus. They searched the genomes of 50 modern species of mammals to identify fossilized sequences of ERBFC and found them embedded in the DNA of over half of those species. Those sequences were even shared between species as distantly related as humans and aardvarks. Now, when a biologist sees this kind of pattern, the first thing they try to figure out is whether there's some simple evolutionary explanation for it. Like, did the virus infect some early mammal that was the common ancestor of many different groups, so that this molecular fossil was just passed down through all of its descendants? In this case, the answer turned out to be no. It's a little more complicated than that. Instead of one infection event, it looks like this virus independently infected different mammal species and embedded itself in their genomes multiple times. For example, 12.
Sorry, I meant to catch that just a little bit earlier, right here. You guys see the difference in those two trees that he just presented? So this one, each one of these red dots indicates where they found the introduction of that viral genome piece, that ERB. You guys follow that? So before this, he put that dot there, right? And then he said maybe if it was inherited here, if that was one infection in one lineage, it was a common ancestor of all of these organisms, that could explain it, right? Why you see that genome, that piece of that viral genome in all of these other organisms, but that is not the case, right? So it's not homology, right? It's not something that happened in this shared lineage that then passed on to everybody, but it happened independently all of those different times. Does that make sense? And looking at the two trees, give me a thumbs up if it makes sense. Very few thumbs. There's a few. Okay. So again, with applications of phylogenetics, right, figuring out where this came from and why it's valuable.
Itself in their genomes multiple times. For example, 12 of the mammals studied, including humans, pandas, and dogs, all have more than one genetically distinct sequence of ERBFC in their genomes. This suggests that there were many independent infection events by different strains of the virus as it evolved while spreading around the world. So we actually have two different fossils of ERBFC in our DNA. And as far as we know, they don't do anything right now. The older one comes from an infection in the common ancestor of all great apes, sometime between about 20 million and 16 million years ago. And the younger one we got from the common ancestor of all African great apes, between about 16 million and 9 million years ago. As for how widespread this pandemic was, it's likely that the virus made it to every continent except Antarctica and Australia, which were geographically isolated at the time. And this may explain why no Australian marsupials in the study, like wallabies and Tasmanian devils, were found to have any of that virus in their genomes. So those are some of the things that we can figure out about this ancient pandemic. But there are other things that we just can't because the fossil record of this virus is incomplete. After all, there are probably species that lost their sequences of ERBFC over time. And others were probably infected, but the virus didn't embed itself in their DNA successfully. And some lineages that might have carried the viral fossil have just gone extinct. So we're missing pieces of the puzzle, which makes some of the details of the virus's spread a mystery. But by using that method known as the molecular clock, and by building a family tree of virus strains across mammal species, researchers think that the virus originated around 33 million years ago. And from there, it spread around the world for at least 15 million years. While we don't know what species the virus infected first, we can tell that it started jumping from group to group pretty quickly. There's evidence of at least 26 independent transmissions between species. And some of these events look pretty weird because the jumps weren't always between closely related species. For example, the sequence of this virus found in dolphin DNA is closest to those found in rodents and rabbits. Now, this doesn't mean that a dolphin caught the virus directly from a rabbit or vice versa, although I personally would love to know what that might look like. Like I said, the fossil record of this virus is incomplete, so we're probably missing some steps there. All we have to go on is what we know about how viruses jump between species today, like by one species preying or scavenging one another. Beyond that, we just don't know. But one of the
Okay, two things. First of all, I want to point out that this is actually a graphic that is called a tangogram. I just think that's cool. You can see why, right? It's like the worst phylogenetic tree you've ever seen because it's all tangled up, yes? It's actually called a tangogram. Did you know that existed? I didn't know that until I watched this video. The other thing is, remember how he just mentioned that the two closely related sequences of this viral ERB are found in rabbit and dolphin, yes? And he said, how could that have happened? He doesn't know, right? What do you guys think? Any hypotheses? Think about what we talked about really early on about Pakicetus. You guys remember Pakicetus? It was like the first week or second week class. So if you don't, that's no big deal. That's one of the evolutionary ancestors of cetaceans, like whales and dolphins. Okay, so this would have happened about 30 million years ago or so. So whales were probably already, and dolphins were already on their way to being what they look like now. So it probably wasn't a wandering predatory mammal ancestor, but something along those lines could be possible. Right, so I want you guys to think about the fact that just because it's in modern day rabbits and modern day dolphins doesn't mean that 30 million years ago, that rabbit looked like a rabbit and that dolphin looks like a dolphin. Does that make sense? So it's always about thinking in terms of those ancestors and those ancestral forms rather than thinking about modern day organisms as we see them. Does that make sense? So maybe the interaction between the rabbit ancestor and the dolphin ancestor was different than what you would see today. Yes? So he goes on to talk about one organism preying on another organism, that sort of thing, and that's how you can sometimes get these host shifts with viruses. But I just wanted to point that out so that you're thinking in evolutionary terms and not necessarily in terms of modern day. And also that there's such a thing as a tangogram.
That's what hopping between species may have been its ability to swap genes with other strains and with other totally different viruses over the course of the outbreak. This is known as viral recombination, and it can happen when two different viruses infect the same animal at the same time. This lets them splice elements of their genomes together as they replicate within the same cell, and it happens naturally all the time. In fact, it looks like this is probably what happened to allow SARS-CoV-2, the virus that causes COVID-19, to spread so successfully. There's good evidence that most of this virus is similar to coronaviruses seen in bats, but a bit of a protein that helps it enter its target cells looks more similar to another virus found in a pangolin. We still don't know what host animal is infected by both viruses, though. Now, recombination can also happen when a virus picks up genes from viruses that had already embedded some of their sequences in the host genome. And there's evidence of multiple recombinations over the history of ERBSC. So it might have gotten the genes that helped it jump between species from other viruses and other strains. And this seems even more likely because one of those recombinations is in the protein that helped the ancient virus enter its target cells, which sounds pretty familiar. But what kind of virus was ERBSC? And how deadly was it? Well, we do know that the virus infected mammals, replicated in their cells, and occasionally became embedded in their genomes. And if you look at its closest living viral relatives, try to figure out what kind of infection it might have caused. Based on this genetic structure, ERBSC is an ancient member of a genus called gamma retroviruses. Today, this group includes the murine leukemia virus in mice and feline leukemia virus in cats. And one of the most common effects of this group of viruses is oncogenesis, or the formation of cancer in their hosts. So it's possible that the ERBSC virus caused similar illnesses, including cancers. But because we don't know what kinds of cells it infected, we can't tell whether it was likely to have caused leukemias or some other kind of cancer. And we might never know. As the genomes of more species are sequenced, our ability to untangle the histories of these viral fossils will only improve. Because ERBSC isn't the only viral fossil in our DNA. There are many others in the human genome whose stories are just waiting to be told. And without the record in our genome, we'd never have known this epic ancient pandemic ever happened. And these ancient remnants don't just help us understand what happened in the deep past. They show us that viruses have jumped between species for millions of years and still do. And they also help us understand and predict how new viruses behave and spread today. which I think we all can agree is pretty important, from one mammal millions of years ago to our DNA today. The story of ERVFC reminds us that pandemics are part of the history of life on our planet. And whether we like it or not, they'll be a part of our future too. Now, while ERVFC is a
The rest of this is all like Excel and stuff, so we're gonna skip that in. But I'm sorry again about the sound, but go back and watch that again if you have some time. You guys will see some questions on your next exam that are over this video. Okay, kind of like you did on the first exam, where I will specifically tell you, hey, these questions come from this video, but you will need to understand it pretty well, okay, so that you know what's going on. It's not one of the bonus ones. But isn't that interesting? So he was talking at the very end about the relevance to the current virus and circulating, and how they think that that sequence that they find in all of our genomes now had to do with a particular protein that allowed the virus to enter the host cell. You guys know what protein that is on coronavirus, on SARS-CoV-2? That's spike, right? That spike protein that connects to the host cell. Remember we talked about viral receptors on host cells and how the virus has to attach something to get in. So the similar type of protein in this ancient virus that was getting into cells that way. So pretty interesting parallels, I think. Yes? All right. That's back to viruses, but it's all relevant. Okay, so don't forget, even though we just finished bacteria, that when you go back to look for that video, it's in the virus chapter. I will remind you guys of that. Maybe I'll add it to your study guide and put it in there for you. All right, exciting stuff. You haven't seen exciting yet, because now we're going to talk about protists. Yeah. I love how excited you guys get about all this stuff. I'm kidding, obviously. Okay. Have you heard the term protist before? Or protista as a kingdom? High school maybe? You've taken biology classes before? You heard of protists? Some of you are nodding. What is a protist? Don't be afraid to be wrong or right. Just guess. Or tell me what you remember. What do you remember? You were nodding vehemently. I'm going to pick on you. Simple organism. In many instances, that is true. So what do you mean by simple?
I remember some of them were autotrophs, and I believe some were also heterotrophs.
Autotrophs and heterotrophs. We'll talk about that. So there's a ton of diversity, okay, in this group. Now, it depends upon your view of phylogenetics where these guys fit. Okay, so they're still lumped together. All of these organisms we're going to talk about as protists are still lumped together in this group. Okay, but we're going to look at some phylogenetic trees, and we're going to talk about the actual relationships between these guys. So this is kind of our most controversial taxonomic group that we'll discuss. And we'll talk about why that is going forward. And this intro slide is just some pictures to get you looking at some of the examples of protists. There's a tremendous amount of diversity, and we're going to talk about why. So rather than saying what is a protist, which I just did to you, so I apologize, because that's not really a fair question. It's easier to ask what aren't protists. Okay, so when you talk about a protist, it's really a group of organisms that have certain things in common, but don't fit in any other group. Okay, so what I mean by that, protists are eukaryotes. So we are now switching gears. So this is a good place to take note that we have finished prokaryotes. That is 2 of the 3 living domains, so we covered them in one chapter, bacteria and archaea. Everything we talk about going forward are eukaryotes. Okay, what is a eukaryote? What does a eukaryote have that a prokaryote does not? Membrane-bound organelles, membrane-bound nucleus. There are other characteristics of eukaryotic cells that we're going to get to in just a few slides that are going to be critical to our discussions going forward, that are going to get a little bit more in depth than what you've learned in previous classes, which is membrane-bound nucleus, membrane-bound organelles. Okay, but we're going to talk more about that. So all organisms that are classified as protists are eukaryotes. Okay, but they don't fit in with plants. They don't fit in with animals, and they don't fit in with fungi, which are the other 3 groups that we're going to talk about, the other 3 kingdoms, okay? So the group Protists, the kingdom Protists, is really a catch-all group for eukaryotic organisms that don't fit in those other groups. Okay, so that's why I say it's more realistic or more fair to ask what aren't they? Okay, they aren't plants, they aren't animals, and they aren't fungi. So they lump them all together into a big pool and call them Protists. We're going to look at some trees, as I mentioned. Recent molecular systematics has revealed that some Protists are more closely related to animals or plants or fungi than they are to each other. So we'll look again at monophyletic groups. Okay, and Protists as a kingdom is not a monophyletic group. What is a monophyletic group? Do you guys remember? Or a clade? Other synonym is a clade. What do you have to define a monophyletic group on a tree? A common ancestor. Yeah. Okay. So we'll talk about why Protists is not a monophyletic group going forward. So most of these guys, most organisms classified as Protists are microscopic. So that gets at Michael's simple comment, right? Small. Many unicellular, but not all. Okay, so you've got some larger scale, visible with the naked eye, macroscopic examples, things like slime molds. Very exciting. We'll look at those in a little bit. And you've got multicellular examples, things like seaweed. Most of the time, if you're at the beach and you reach down and you feel something brushing on your leg, you pull it out of the water and it's seaweed, that's probably a Protist. But it's not unicellular, right? It's not this one teeny tiny microscopic thing. It's a big old blob of seaweed, right? So that's an example that doesn't fit the mold. Because Protists are this catch-all group, as I'm calling it, it's not surprising that there's so much diversity in Protists. Okay, so we're going to look at examples from all of the 6 major subdivisions of Protists, and they're going to look really different from one another. They're going to have differences in metabolism and lifestyle and size and shape and form, right? So just keep that in mind, that there's a ton of diversity in this group, but it's because they're not really all related to each other. It's this catch-all group that doesn't fit anywhere else. All right, so just keep that in mind going forward. And this is the only group that behaves this way, so sort of unique in that sense as well. Okay, so just keep that in mind. All right, this is where it gets really fun. All right, this topic right here, the origin of eukaryotic cells, is where we're headed with our first class discussion, our first official class discussion. I know, you cannot wait. So I'll show you guys in just a minute after we finish talking about this slide, where those materials are for that class discussion and what I want you to do. So we talked about the age of prokaryotes, right? We said 3.5 billion years ago, maybe further back even, right, for the earliest prokaryotes. We said cyanobacteria about 2.4 billion years ago, right? So really old. When we look for the first eukaryotic cells in the fossil record, they're about 2.1 billion years old. Okay, so not too far beyond the evolution of photosynthesis. Okay, so just something for the timeline there. But where did they come from? How did we get from prokaryotes, which are simple, single celled, no nucleus, reproduced by binary fission, sort of extremes, living in different extremes of atmospheres? How did we get from that to eukaryotes? Where did they come from? This has been a burning question in evolutionary biology for hundreds of years. All right, so what we're going to talk about in our first discussion and going forward here quite a bit is the endosymbiotic theory, sometimes called endosymbiosis, sometimes referred to as the serial endosymbiont theory, SET. Okay, we're going to talk about why. But this is a theory of the origin of eukaryotic cells. Where did these complex cells come from? But here's what I want to talk about first. What is a theory in science? Is it an educated guess? It's not, right? That'd be a hypothesis. Is it like what you think might have happened, like a prediction? It's not. Tell me what a theory is. What do you have to call something a theory? Cell theory, germ theory, evolutionary theory, endosymbiotic theory. What's a theory? It's not an educated guess. Is it stronger than that or weaker than that? Stronger, yeah. There's more what? Evidence. Absolutely. So a theory in science, it's still not necessarily immutable. meaning it can change, right? New evidence can always adjust the way that we understand things, but a theory is an overarching explanation, right, for a particular phenomenon or set of phenomena that have been experimentally investigated over and over and over, okay? So remember how we talked about that when we were talking about evolutionary theory. Yes, evolution is a theory because it has been confirmed through scientific evidence over and over and over again. Same thing with this, okay? So this is not just a wild guess. It sort of started out that way, okay? So over 100 years ago, there were scientists who were making these hypotheses that perhaps evolution of eukaryotes occurred because one prokaryote, and bear with me, you guys, ate another one, okay? Pac-Man style. Have you guys heard of endosymbiotic theory before, or is this brand new? Brand new? Heard of it before? Okay, it's in there. All right, so the theory itself, just to sort of summarize quickly, states that eukaryotes are the product of one cell engulfing another, living symbiotically together, close association, and evolving together over time until they were no longer recognizable as separate cells. One eats the other or engulfs the other. So I'm using the term eat loosely. Okay, so it could have been just a merging of cell membranes potentially, right? We don't really know how it happens because none of us were around to see it. But what we do know is that organelles that we find inside of eukaryotic cells, things like mitochondria, what does mitochondria do? ATP factories, right? Powerhouse of the cell. Everybody learns that in middle school. Things like plastids in photosynthetic plants, like chloroplasts. What does a chloroplast do? What is the main responsibility of a chloroplast in a photosynthetic organism? The chlorophyll's in there because what happens? What are you using chlorophyll for? Photosynthesis. Yeah. Yeah, so yes and no. Photosynthesis, yeah. So you got mitochondria, which is the site of ATP synthesis in eukaryotes. You've got chloroplast, which is the site of sugar synthesis, essentially, photosynthesis in eukaryotes. Those organelles came from previously, historically, billions of years ago, free living prokaryotes that were able to do those things alone. Okay, so we're talking about aerobic bacteria. Bacterial cells that can use oxygen to make ATP, okay? And we're talking about things like cyanobacteria, photosynthetic microorganisms, that are then engulfed by other prokaryotes, start to live together, that's where this living symbiotically piece comes in, and are so successful at doing that, that relationship persists from one generation to the next until eventually they become one organism. So this is not something that happened overnight or in one generation, but in many, many generations of reproduction. But think about how quickly microbes reproduce. How quickly can they do that? Fast or slow? Fast, right? You watch the video of antibiotic resistance evolving in a population, and it's on the order of hours to days for prokaryotes to reproduce. So if you think about millions of years, 300 million years or so, that's a lot of generations, right, of prokaryotic reproduction. So it happened over time until ultimately they lived together, not recognizable as separate cells, okay? I definitely want to spend a good amount of time talking about this, but that's what the class discussion is going to be for, okay? So that's going to be next Monday. So you have a week to read the stuff I want you to read. You have a set of questions that you're going to answer based on those readings, and then we're going to come back and we're going to talk about it on Monday, okay? And because I know how much you guys love to talk voluntarily, I'm going to actually assign each of you a part that you're going to be responsible for leading the discussion on, okay? Now you're all like horrified. Oh God, you're going to make us say something. But there will be multiple people with the same assigned sort of area, so you're not all alone, okay? But you are going to be called upon to sort of start the discussion on those particular questions, okay? So that stuff is up. I'm going to show you really quickly. Before I show you in D2L where that stuff is, I want you to read your book, It is rare that I give you a specific reading assignment out of your textbook, yes? This is probably the first one this whole semester where I said specifically you need to read this. This is background on the theory of endosymbiosis. It will help you understand the other readings that I'm going to give you, which are pretty short. This is also short. This is chapter 23.1. It's just the 0.1 part. Okay, you don't have to read the whole chapter, but you need to read that intro. That'll be really helpful. The next slide in the PowerPoint is a PBS Eons video by the same people who we just watched the virus video on. Really good stuff. It's all about serial endosymbiote theory. Okay, so watch the video. Read through the reading, watch the video. I'm not going to play it now because we already watched the video together. What I want to show you, though, is back over to D2L. There's that new tab that I was showing you about called, you guessed it, class discussions. So if you go in there now, there's a folder called endosymbiosis. That's what our discussion is about. Your required readings are right here. There are 3 of them. Okay. This first one from Berkeley's evolution department is the longest one, but it is written for students of evolutionary biology. It's not a scientific paper, so it's a little bit longer, but it's more like an article. Okay, like not a science article, but I don't know, it's kind of like the one we read about butterfly wings that was published in the Atlantic, like a popular type writing, so it's easier to read. These 2 are both from Nature, but they're like press releases, so they're super short, like a couple paragraphs each. Okay, so this is not a ton of reading, so don't, again, freak out about that. But you'll read those 3 things, and then you have discussion questions, and there are 9 of them, and everybody must answer all 9. Okay. These are based on your readings, okay? So they don't have to be, like, it's not a test, all right? I'm not gonna go in and grade you and say, well, this was a little bit wrong, so you lose points. It's a matter of participation in this discussion activity, okay? So this is part of your participation grade. It's 30 points out of the 100. Half of it comes from turning these questions in, and the other half comes from being an active participant in the class discussion. That's the part that I'll assign to you guys coming forward, okay? So I'll post those assignments on D2L. Anyway, in the meantime, before that, you're answering all the questions anyway, so that's where you find them. So far, so good. Textbook, video, and then these 3 required readings that are posted on D2L. When you have the questions done, there is a folder called Endosymbiosis Discussion Questions. How about that? In your assignments module, where you will upload those. And you'll notice the due date is 11 a.m. February 22nd. When is that? When is the 22nd? Next what? Monday. Yeah, next Monday. So we're having the discussion Monday. What is it? Why is 11 a.m. significant? Yeah, before class, because we're having the discussion in class. So I want to make sure that you have your questions uploaded before the class starts. Yes? Yeah. Okay, there's the plan for that. It's actually, I know this sounds so exciting and fun, but I promise you that it is. It's actually a really good exercise to sort of get into this theory a little bit, because it is notoriously hard to wrap your brain around something like this, right? For anyone who's reading and learning about endosymbiote theory, it's kind of like, how does this happen, right? But it's really, really important that we get a good understanding of this, because this is a significant area of ongoing research in biology. And it is the underpinning theory that leads us to eukaryotic evolution, okay? And you're a eukaryote, right? Plants are eukaryotes, fungi are eukaryotes. This is a significant event in the evolution of life on the planet. So we really need to get a good understanding of it. And this set of readings will help us do that in our discussion as well. So that's the sort of rationale behind why we're gonna do this. You guys good? You up for it? I know I'm gonna make you talk, it's terrible. So mean. Okay, this I think is the last thing I want to talk about today. And then we'll sort of move on from here. Next time, talking about the phylogenetics of the group and the relatedness of these groups within Protists. But this is what we're talking about. There's a lot to unpack on this slide. So yeah, we've got 15 minutes left, but it'll probably take us that long to talk about all of this if we're close to it. So there's a ton of information here. You guys need to know this. Okay, this is not the kind of slide where there's like one question on the exam that you might say vocabulary words. This is also fundamental information about eukaryotic origins. It's the characteristics that are common to all eukaryotes. Okay, So when we're talking about endosymbiosis as the origin of these cells, we're talking about that event leading to this complexity that makes eukaryotes eukaryotes. Okay, that makes eukaryotic organisms so much more complex than their prokaryotic ancestors. And so this stuff is significant too. And although there's a lot here, some of it is review. Okay, so that's great. We're still building, right? We're building on your previous knowledge. So these are characteristics that were present, as best we know, based on evidence, in the common ancestor of all eukaryotes. Okay? Remember when we talked about the last universal common ancestor? We've been sort of using an acronym. What's the acronym for the last universal common ancestor? LUCA, right? You guys on Zoom see this? There we go. All right, so guess what the acronym for the last eukaryotic common ancestor is? LUCA. Okay, so there's an acronym for that too. So this is last universal common ancestor. That's at the root of the tree that has everyone. And this is LUCA, last eukaryotic common ancestor. Okay, so there's a term for that too. Just be aware of the differences. While I'm thinking about it, those of you who are listening in on Zoom, if you're in the web-based course, the discussion assignment for you is different. So look on D2L for description of your particular version of that discussion, because it isn't the same. So don't panic. Your readings are the same, so you can get started, but don't necessarily do anything yet. Okay, I'll post an announcement later today about that for you guys. Okay, so we're talking about the common ancestor, or LUCA, of all eukaryotes. And the evidence that we have is that that each of these characteristics are present in each major lineage of extant eukaryotes. Okay, that's a lot of verbiage there. What does that mean? What about the term extant? You guys comfortable with that? You know what that means? Do you know what extinct means? You do, right? What does extinct mean? It's not around anymore. So extant is the opposite of extinct. Okay, so what do you think extant means? They are currently alive, yes. So when we talk about extant groups, we're talking about those that are currently living, that you can find an example of if you go outside and look for it, or in your guts, whatever, right? They're on the earth, okay? Currently alive. So these characteristics were present in LUCA as evidenced by their presence in each major lineage of extant eukaryotes. So eukaryotes that exist today, whatever group they fall into, and I'll give you a little preview of the tree. Okay, here's the tree of the 6 major groups that we're going to discuss. All right, so members of each of these major groups of protists all have these characteristics. Okay, so the evidence is for homology, that LUCA had these, and that is why you see this in each of these extant lineages. You guys follow? Okay. When we get to number 8, it's a little dicey, whether that's homology or convergent evolution, and we'll talk about that. But we'll start with number 1, which is solid, okay? And also review. The nucleus. What is the nucleus? Central organelle, what's in there? Genetic material, your DNA, right? What makes it special? What does it possess, wrapping that DNA? The nucleus hat. Nuclear envelope, yeah, we're calling it membrane-bound nucleus, right? So there's that nuclear envelope with nuclear pores. You guys remember what nuclear pores are? Basically, pores are holes, yes? So things can come and go, namely messenger RNA, right? For one good example. So you've got your membrane-bound nucleus. All eukaryotes have it. We know that. We've known that since, I don't know, 4th grade maybe. You first learned about cell biology. Maybe late, maybe 7th grade, I'm not sure. But, you know, it's been a while. So you've known about the nucleus. And then mitochondria, powerhouse of the cell, right? ATP factory. All eukaryotes have them. But some are remnant. Remnant is another synonym for something like vestigial. You guys remember what vestigial structures are? Like whales with pelvic bones. What does vestigial mean? They aren't common ancestor. Yeah, it's been there a while. It's evidence of homology, right? But it is bits and pieces. It doesn't necessarily have a function, but it used to, okay? So most extant eukaryotes have recognizable mitochondria that are being used for cellular respiration, production of ATP using oxygen, and so on and so forth. But there are some examples of eukaryote protists, right? All protists are eukaryotes, so that's redundant, but protists that live as parasites. And those that live as parasites may have lost functional mitochondria because they don't need them anymore. Okay, but that is a secondary loss. Okay, that would be the same thing as a snake losing its legs or a whale losing its hind limbs. Common ancestor had it, but over evolutionary time, that particular group lost that trait because they don't use it. No selective pressure to keep it, okay? So most of the time you can find the mitochondria in a eukaryotic cell. Sometimes you find evidence of mitochondria. It might be a piece. It might be a piece of the cell membrane that you can identify. It might even be genes that code for the production of it. Okay, so it might be a very trace remnant, but it's there. Okay, so that's a shared characteristic. Cytoskeletal elements, number 3, things like actin, microfilaments, microtubules, centrioles, those things that move things around inside of the cell. You guys probably remember that from mitosis. Those are unique to eukaryotes. Prokaryotes don't have those cytoskeletal elements. This will be a topic of discussion in that Berkeley article on endosymbiont theory. Okay, so you'll read more about cytoskeletal elements and where they came from, hypothetically, in your assigned reading. Flagella and cilia. We talked about flagella. What are those? Little tails, right? That whip to push you around for motility. And cilia are usually present in multiple numbers, right? Cytoplasmic extensions for things like attachment, sometimes movement, sometimes feeding, motility as well. So most groups, most eukaryotes have flagella and or cilia. If you don't have them, you have a common ancestor with them. Okay, and we're not talking about LICA, we're talking about a common ancestor in the group that you belong to. So when we get to these next time, on Wednesday, we'll talk more about that. But say, for example, humans only have one type of flagellated cell, sperm cells, right? But if you go back a little bit further in this group, in the opisthecons, where animals fall, you've got flagellates that always have a flagellum. So the idea is a more recent common ancestor with flagellum. So if you don't have it, again, that's a secondary loss. You guys with me on that? Okay. All right, how are we doing? 5 minutes. Can we do it? I think we can. Chromosomes. We talked about how eukaryotes have linear DNA, as opposed to what in prokaryotes? What do their chromosomes look like? Circular. And how many do they have? 1. What about eukaryotes? Linear and what? How many? More than 1 in just about every case. And also we have histones, which are those proteins, right, that the chromosomes, chromatin strands wrap around as they're condensing into chromosomes before mitosis. We talked about those briefly. Mainly the chromosome structure is different, so just know that, okay? Mitosis is nuclear cell division. You guys all learned prophase, metaphase, anaphase, telophase, cytokinesis, right? If you don't have a nucleus, you don't have to do that. We talked about binary fission, yes, in prokaryotes. You don't have to replicate your nucleus and make 2 new ones before you can do cell division if you don't have a nucleus. You guys follow what I'm saying here? Picking up what I'm putting down? If you don't have a nucleus, you don't have to do mitosis, you just do binary fission. So mitosis is unique to eukaryotes. And sexual reproduction. Genetic recombination as a result of meiosis and karyogamy. Okay, that is a eukaryotic thing. Again, prokaryotes use binary fission. Okay, so when you're talking about genetic recombination, what's meiosis? It's cell division. What does it make? What's the end result of meiosis? What type of cell? Haploid gametes, right? Yeah, so then you get recombination when they refuse. You guys know this term, karyogamy? I don't think we've used this term yet. What is karyote? Remember when we talked about the root word for eukaryote versus prokaryote? And I told you karyo means kernel or seed, but what does it refer to? Nucleus, yeah. And then gamete is like fusion, okay? So when you think about a term like monogamy, it's like marriage, right? When you get married, that's a monogamous relationship, or you're in a committed relationship, it's like one fusion, right? It basically means put together. So karyogamy is nuclear fusion. So meiosis results in haploid gametes, sperm cells and egg cells. When they recombine, the first thing that happens is those cell membranes fuse, and then the two haploid nuclei fuse to make that first diploid nucleus that results in the zygote. You guys follow? This is stuff we've talked about. So karyogamy is that fusion of the two haploid nuclei, recombining the genetic material from the two parents. So that's sexual reproduction in a nutshell, unique to eukaryotes. All right, really quickly, in the last couple of minutes, let's look at cell walls. What are some examples of organisms that have cell walls? We talked about gram-negative versus gram-positive, right? In peptidoglycan, who has that? We just talked about it. Bacterial cell walls, right? Prokaryotic cell walls. What about plants? You guys remember learning about plant cell walls? I know that's been a while too in the early chapters from Bio 1. Fungi, most fungi have cell walls. But we don't. Animal cells don't have cell walls, right? We just have a cell membrane. We don't have that extra structural sort of supportive layer. So all major lineages, when we talk about those lineages, we're talking about these 6 major groups that we'll go over on Wednesday. There are members of each of these 6 major groups of protists that have cell walls, but there are also members of each of those major groups that do not have cell walls. So it kind of goes against, remember I said this is where it gets a little dicey, it goes against the statement that that the common ancestor of all eukaryotes have this, because with the cell wall, there's evidence that suggests it could be homology, but there's also evidence that suggests that it's convergent evolution, meaning if you have a cell wall, you evolved it as a derived trait. You guys with me on that? Derived trait. Or that everybody had them and it was homology, but that you don't have it now as a secondary loss. So that is not conclusive. You guys okay? I know that's a lot. Like I said, 15 minutes on a single slide is a lot to unpack. We will revisit this on Wednesday before we jump into the phylogeny of protists and start talking about these groups. After that, after the tree, we will start talking about notable protists from each of the groups, and that's where it gets really interesting, because let me tell you guys, these dudes do some weird stuff. Okay, so that's the fun part. You just start talking about the weirdos and the interesting examples. So that's where we're headed.